So in tumor cells, the glycocalyx that Greg just described is highly enriched. This characteristic is used in detection of cancer, but the influence of this envelope was still very poorly understood. The study started when the group of Valerie Weaver demonstrated that large glycoproteins, including the mucin one, are abundantly expressed, especially by circulating tumor cells from patients with an advanced state of the disease. They showed that those large glycoproteins regulate the nanotopology of the cell surface. Until our study, it was thought that a bulky glycocalyx was preventing cell adhesion through integrins, because integrins are 10 times smaller than the glycocalyx thickness. Counterintuitively, the group of Valerie discovered that this large glycocalyx triggers the formation of more and bigger adhesion sites. To understand this glycocalyx paradox at the molecular level, they needed to have access to the dynamics of individual proteins such as mucin and integrins. And that's why they came to us. At that time, we had published a study in Nature Cell Biology where we used super-resolution techniques, microscopies, and single protein tracking to study how integrins were activated. Indeed, Bordeaux is the place because it's an open pole node for the development of super-resolution microscopies and single protein tracking. Using such microscopy, we can produce such movie in which, in fact, we are able to follow here in red individual integrins diffusing around and stopping inside adhesion sites here in green. In doing so, we deciphered the crucial molecular events leading to integrin activation, which means integrin immobilization. And that's done by its interaction with its main partner called tannin. For this present study, we followed mucin proteins itself and showed that mucin was diffusing fast but cannot access inside adhesion sites. When we followed integrins, we observed that the presence of the mucin did not modify the diffusion of integrins, but prevented activated integrins to get immobilized except in the already existing adhesion sites. This study elucidates the molecular mechanism by which the glycocalyx controls integrin activation. In this model, the large envelope of glycoproteins, here in red, funnels the integrin activation, here in green, in already existing adhesive areas. This glycocalyx generates a mechanical force that will activate integrins and thus trigger signals for survival and proliferation of cancer cells, and that even on soft environments where normal cells don't grow. Thus, the expression of large glycoproteins by cancer cells give them the ability to create their own mechanical environment, leading to the activation of integrins and invasion of the cancerous cells throughout the body. This multidisciplinary study at different levels from the molecule to the body and using medicine, clinical chemistry, cell biology and biophysics has revealed how cancer cells hijack the normal regulation of the cell and use the glycocalyx to survive and proliferate in unsuitable environments. The result of this research will open avenues for new therapeutic strategies targeting protein glycocalyx to halt the onset of metastasis and the spread of cancer.